Welcome to my channel, Deep Learning Biology. Okay, so in this video we are going to solve more numerical problems related to DNA RNA quantification. Hopefully you must have watched our first video, DNA RNA quantification first video. Now this is the second video, and here I just want to remind you, keep in mind, for double stranded DNA, okay, 1 OD, it is equal to 50 microgram per ml. Okay, for single stranded DNA, okay, 1 OD, it is equal to 33 microgram per ml. Okay, and for single stranded RNA, keep in mind, 1 OD, it is equal to 40 microgram per ml. Okay, so these relationship we are going to use to quantify DNA. If it is double stranded DNA, keep in mind 1 absorbance, okay, 1 OD, it is equal to 50 microgram per ml. For single stranded DNA, 1 OD, it is going to represent 33 microgram per ml DNA concentration. For single stranded RNA, 1 OD is equal to 40 microgram per ml. Now here I also want to tell you, so whenever you have to calculate, okay, try to see here, we are going to have a stock of DNA. Okay, from this stock, okay, you will take few microliters, okay, and rest you will add water. Okay, most of the times we are going to make the final volume 1 ml, and by using this sample 1 ml, okay, we use a quartz cuvette. Okay, then we are going to see the whatever the value of absorbance we are going to see within the spectrophotometer. Okay, so what is the OD value? Now here I have told you, for double stranded DNA, okay, 1 OD, it is equal to 50 microgram per ml. Okay, now try to see, let's suppose here this is your sample, okay, and here you are getting the value 1. Okay, now this is going to represent the concentration which is present within the cuvette. Okay, so this is going to represent the concentration which is present within the cuvette, not of the stock. But try to see, we are interested in calculating the concentration of DNA which is present within the stock. So here keep in mind, here concentration of DNA, it is being calculated. Okay, it is being calculated. What is the value of absorbance at 260 nanometer multiplied by dilution factor multiplied by 50 microgram per ml. Okay, you can calculate the concentration here. Okay, it will be absorbance at 260 nanometer, means what is the OD value, multiplied by the dilution factor multiplied by the 50 microgram per ml. Okay, now let us solve some of the numerical problems. Everything will be clear to you. Okay, now write down the numerical problem. From a small culture, okay, you purified DNA, you resuspended DNA in a volume of 50 microliter, you diluted 20 microliter of purified DNA in a total volume of 1000 microliter, and here when you measure the absorbance as 260 nanometer, it is 0.550. Absorbance at 280 nanometer, it is 0.324. And now here calculate. The question 1 is you have to calculate what is DNA concentration in 50 microliter sample. Question 2, how much total DNA purified? And the question 3, what is purity of DNA? So here try to see, from a small culture you purified DNA, you resuspended DNA in a volume of 50 microliter, you diluted 20 microliter of purified DNA in a total volume of 1000 microliter, and here absorbance you calculate at 260 nanometer is 0.550, at 280 nanometer 0.324. Here you have to calculate what is the DNA concentration in 50 micrometer sample, how much total DNA was purified, and what is the purity of DNA. Now try to see here. Here try to understand, you are having a small culture, and from this culture you purified the, here it is double stranded DNA. Please mention it is double stranded DNA. So you have purified the double stranded DNA. Now here initially your DNA is in form of a pellet, and you dissolve this pellet in a volume of 50 microliter. You see, you diluted, you resuspended the DNA in a volume of 50 microliter. Now from here you took 20 microliter, and you add rest amount of water, and you here 980 microliter, and you make the volume, that is 1 ml 1000 microliter. So here now you are going to have 30 microliter. So 20 microliter you are using for quantification. Using a quartz cuvette, you quantify, and then you measure the absorbance at 260 nanometer absorbance. What you find? The answer 0.55. Spectrophotometric readings, they were showing the absorbance at 260 0.550, and absorbance at 280 0.324. Now you have to calculate what is the concentration here. How much total DNA was purified, and you have to comment on the purity of DNA. Now try to see here. Hopefully you remember this relation. Absorbance at 260 nanometer, it is, if you are going to have 1 OD, then it is 50 microgram per ml. Now here keep in mind, this is going to tell you the concentration of DNA which is present within the cuvette. If you want to calculate concentration of DNA of the stock, okay, of 50 microliter sample here, okay, it can be easily calculated. What is the value of absorbance at 260 multiplied by dilution factor multiplied by 50 microgram per ml. Now, what was the reading here? Absorbance at 260, 0.550. Now, dilution factor. Okay, try to see here. What is the final volume? 1000 microliter. You diluted 20 microliter and you are making final volume 1000. So here 1000 divided by 20. This is the dilution factor multiplied by 50 microgram per ml. Okay, if you are going to solve this, it will come out 1375, okay, microgram per ml. Yes, here you can convert into microliter. Okay, 1 ml is going to have 1000 microliter. Okay, so if you are going to solve this, it will be 1.375 microgram per microliter. So this is the concentration of DNA, okay, in your stock, the 50 microliter sample. Okay, now you calculated, okay, in the stock, okay, the 50 microliter sample, okay, concentration was 1.375 microgram per microliter. Means 1 microliter of this sample is going to have 1.375 microgram double stranded DNA. What will be present in 50 microliter? It's simple, 50 into 1.375. If you calculate this, it will be 68.75 microgram. Okay, so we have already calculated what is DNA concentration of 50 microliter sample. Okay, this. Now, how much total DNA was purified? So this is your answer. So here total DNA, okay, purified was 68.75 microgram. Now you can also calculate what the total DNA you have. Try to see here you have used, okay, 20 microliter for quantification. Now how much you are going to have? Now 30 microliter. Okay, so here you can also calculate, okay, 1 minute please. Okay, so now you are having a stock 50 microliter minus 20 microliter. 20 microliter you have used for quantification. You are now having 30 microliter. Okay, now here this 30 microliter is going to have DNA. Very simple. 1 microliter is going to have 1.375 microgram. Okay, 30 microliter will have 30 into 1.375. Okay, so here if you are going to solve, it will come out around 40.5 microgram. Okay, this amount of DNA you have. Now there is another, what is purity of DNA? Now hopefully you remember, okay, to check the purity of DNA, what we do, okay, we calculate 260 by 280 ratio. Okay, here you see absorbance at 260. What is being mentioned? 0.550. Absorbance at 280. Okay, it is 0.324. Okay, absorbance at 260 and absorbance at 280. Okay, now try to calculate. Okay, you use your calculator and try to calculate. Okay, so here you have to just divide. Okay, 0.550 divided by 0.324. Okay, so here it comes out 1.69. Okay, so here if your answer is above than 1.5, then you can say your DNA is pure. Okay, you can do some experiments with this DNA. Okay, so here you see the ratio is greater than 1.5. Okay, then you can use this DNA. Okay, it is going to have less amount of proteins. Okay, so your DNA is more pure. Just remember the ratio must be more than 1.5. Okay, so hopefully you have learned this. Okay, now let us try to solve another numerical problem. Very simple. Okay, single stranded DNA of length 7250 nucleotide length. Okay, now we are going to have single stranded DNA and length here is 7250 nucleotide. Okay, is diluted, okay, 10 microliter into a final volume of 1000 microliter. The absorbance 260 value, okay, it is being mentioned. It is now 0.125. Okay, what is the concentration of DNA in microgram per ml? Okay, and here you have to also express answer in picomoles per microliter. Okay, so single stranded DNA, length 7250 nucleotide. It was being diluted 10 microliter in a final volume of 1000 microliter. What you get the absorbance, the OD value 0.125. What is the concentration of DNA in microgram per ml? Express answer in picomoles per microliter. Now try to see here. Here you see you are having single stranded DNA. Hopefully remember 1 OD for single stranded DNA 33 microgram per ml. Okay, for double stranded DNA it is 50 microgram per ml. For single stranded DNA it is 33 microgram per ml. Now you are having a stock. Okay, from this stock you take out 10 microliter, okay, and you diluted it to 1000. Okay, so what is the dilution factor here? It is 1000 divided by 10. You have diluted it 100 times. Okay, and here when you calculated the absorbance, okay, absorbance at 260, it is 0.125. Okay, now you can calculate the concentration of DNA. Concentration of DNA, it is equal to absorbance at 260 multiplied by dilution factor. Okay, absorbance at 260 multiplied by dilution factor and multiplied by this 33 microgram per ml. Okay, so let us solve here. Here concentration of DNA, it is equal to, here it is single stranded DNA, it is equal to 33 microgram per ml multiplied by dilution factor multiplied by absorbance value at 260 nanometer. Here it will come out 33 multiplied, what is dilution factor? 1000 divided by 10, and here absorbance 260, it is 0.125. Your answer will come, if you are going to solve this, 412.5 microgram per ml. Okay. Now there is another thing here. If you see in the last, the question is express your answer in picomoles per microliter. Picomoles, okay, per microliter. Okay, so you have to convert this answer in molar answer. Okay, it is simple. Okay, just you have to learn, hopefully you know, okay, molecular weight of double stranded DNA. It is equal to 660 gram per mole. Keep in mind, molecular weight of double stranded DNA, it is 660 gram per mole. And if you see molecular weight of single stranded DNA, it is half, 330 gram per mole. Hopefully you know, 1 mole of any substance is going to have 6.023 into 10 to the power 23 molecules. Try to see here, double stranded DNA. Okay, let's take one nucleotide pair. A is paired with T. Okay, now in DNA you are going to have nucleotides arranged. Okay, now here if you are going to see, okay, let's suppose you are going to have nucleotide pairs, and how many? 6.023 into 10 to the power 23, like this. Okay, oh, and if you are going to weigh, okay, so here this has been calculated in taking consideration the average molecular weight. If you are going to calculate, it will come out 660 gram. Okay, so here 1 mole means 6.023 into 10 to the power 23. Okay, base pairs, they are going to weigh 660 gram. Okay, if it is double stranded DNA, and if it is single stranded DNA, it is going to weigh 1 mole of, okay, here for double stranded DNA. For single stranded DNA, keep in mind, 1 mole it is going to weigh 330 gram. Okay, now try to see here. Here, 1 minute please. Okay, try to see here, 1 nucleotide pair. Okay, sorry, here we are having single stranded DNA. 1 nucleotide, okay, it is going to weigh, very simple. Here try to see, for single stranded DNA, 1 mole it is going to weigh 330 gram. 1 nucleotide, okay, it is going to weigh 330 gram per mole. This is the molecular weight. Okay, now here you are having 7250 nucleotide. Okay, your DNA, it is going to have 7250 nucleotide. Okay, so you are going to have nucleotides. Okay, how many? 7250 nucleotides. Okay, now here you see, consider each one as separate. Okay, 7250 nucleotide, they are going to weigh 7250 multiplied by 330 gram per mole. Okay, so here 1 nucleotide, it is going to have weight 330 gram per mole. 7250 nucleotide, they are going to have 7250 multiplied by 330 gram per mole. Now convert this. Okay, so here you see 330 into 7250 Okay, convert gram into microgram. It is just multiplied by 10 to the power 6. Okay, gram. Mole, 1 mole into picomole. Very simple, 10 to the power 12 picomole. Okay, so here if you are going to solve this, it will come out 2.39 microgram per picomole. 2.39 microgram per picomole. Now here try to see. Okay, here you are going to have 7250 nucleotide. Okay, DNA. Okay, single. 1 picomole of this. Okay, 1 picomole of this, it is going to weigh 2.39 microgram. Try to see what is your answer. 2.39 microgram per picomole. Okay, so here if you are going to have a single stranded DNA, 7250 nucleotide length, 1 picomole of this, it is going to weigh 2.39 microgram. Now try to see here. We have calculated the concentration of DNA, 412.5 microgram per ml. Okay, so here you see 2.39 microgram. Okay, it is going to have 1 picomole DNA. Now here you are going to have, let's suppose you are going to have 412.5 microgram per ml DNA. Okay, so 2.3 microgram is 1 picomole. Okay, 412.5 microgram. Okay, what it will be? Okay, very simple. Okay, try to see here. 1 microgram will be 1 divided by 2.39 picomoles. Okay, so here 2.39 microgram is 1 picomole. 1 microgram is equal to 1 divided by 2.39 picomoles. Now try to see here. 412, this was our answer. Okay, we have to express the answer in picomoles per microliter. So just try to convert this. 412.5 microgram means, okay, just 1 microgram this, and 412.5 microgram, 412.5 divided by 2.39, okay, picomole. It will be picomole. Then here you are going to have ml. Okay, just multiplied here, divided by 1000 microliter. So here 1 ml is going to have 1000 microliter. Okay, so if you are going to solve this, 412.5 divided by 2.39 divided by 1000, okay, you will get the answer. Okay, and this answer will be in picomoles per microliter. And if you are going to solve this, okay, it will come out 0.172 picomoles per microliter. Okay, so here you see 412.5 microgram per ml. Okay, and here keep in mind this is single stranded DNA and length 7250 nucleotide. Okay, so here concentration initially we calculated in microgram per ml. Okay, and then we calculated in picomoles per microliter, just by taking the relation that 1 mole of single stranded DNA, it is going to weigh 330 gram. Okay, so hopefully you have understood this. Please do like, share the video and subscribe to my channel Deep Learning Biology. Bye bye, take care.